Okay, so as everyone has taken their seats, I'm going to introduce our next speaker, Francesco Gualdi, who is a PhD candidate, also a member of the Discovery project. Francesco's main interests are the integrating heterogeneous data and translational medicine, also discovering links between genotype and phenotype, and unraveling the role of genes in the development of complex diseases. Francesco finished his bachelor's degree in University of Ferrara in 2017, where he already focused on characterizing the cells of the intervertebral disc. He also did a master's and graduated from the University of Verona in 2020. There, his master's project was in structural biology, and he developed a data set of human G-protein coupled receptors. So currently, Francesco is going to give us a presentation which will be focused on exploring novel biomarkers and especially biomarker tools for the discovery. So the stage is yours, Francesco.
Thank you. Thank you for the kind introduction. Say, probably you remember my CV better than me. So thanks for reminding me. I'm Francesco Gualdi. Let me start to say that I'm honored to be here and talk after Natasha, who gave a beautiful presentation, very interesting for me. And I'm very glad to be here at the Sun Barcelona Summer School, which was important for my curriculum and my career in general. And I'm glad to present my work. I'm studying intervertebral disc degeneration that probably you may have heard of it, like from the previous presentation. So we'll cover it very quickly. So during this degeneration, we have a shift in the catabolism, from the anabolism to the catabolism in the disc. So we have an imbalance of proteins that start to be synthesized that are catabolic proteins. So we have metalloproteinases and a downshift in the regulation, in the synthesization of proteins that are anabolic, such as collagen and aggrecan. Then we have other happenings, such as like the disc is drying. We have a formation, bone formation called osteophytes. And we have signal changes from the MRIs, so we can. It's a very complex condition made of a lot of phenotypes. So my project is to discover, and the topic of the talk is how do we understand a connection between the genetics and then how this is reflected to our downstream pathways, to images, to something that we can measure. And something that we can measure are biomarkers. And biomarkers itself, it's a super huge topic because we can have a different type of biomarker. We can have proteins that are expressed in the disc itself, so we can have like the tissue. We need to measure it in lab. We can find, okay, this protein is more expressed, less expressed, etc. We can have systemic biomarkers. I'm glad I have the pointer, super nice. First time ever for me with the pointer. So we have systemic biomarkers that are protein that we can find in the blood, and they're very cheap to measure, so this would be our preferred biomarkers. But there are some studies in that, and intervertebral disc degeneration is very hard to find proteins that are circulating and reflect the disease. And finally, we can have image biomarkers, so we can measure the disc, we can see it, we can see if it degenerated or not, and so far these one are the best way to go for a disc. But also we have genetic biomarkers, so we can have genetic variants that can tell us, okay, you could develop a disease with that specific risk. We had a beautiful presentation of that from Roger on Monday, that he's studying the genetic predisposition in intervertebral disc degeneration. But we have sorry, yeah? No? Okay. So he talked about genome-wide association studies. That it's our tool to detect genetic biomarkers in complex diseases. So very quickly, there's a control group, so a healthy group that is compared to a disease group. Both of these groups have the genome sequenced, and then we, through linear regression models or logistic regression models, we try to associate variants to the disease, so the disease group. So we see if in the disease group there are enriched variants that could be then related to the disease. This is very linear, very simple, but the reality is different because there are many challenges that we face when we interpret the result, because a logistic or a linear regression gives us a p-value that is a statistical association. So we don't know the mechanistical ways in which the variant then is leading to the disease. So, and we have problems that in the study itself, so in the framing of the study, such as the sample size, because we need persons that have this condition, and many times it has to be big, because we are testing a lot of variants, and so we have to use very low p-value. And we have the phenotype definition as a problem, because how would you define this degeneration? There's a whole environment in research that tries to define the disease itself. And also, these problems are reflected in the missing heritability. It is our variants that we find through the genome-wide association studies are not reflecting the heritability calculated for this degeneration from family or twin studies. So there's something missing. But we have problems also in the interpretation of the result, because when we associate a variant, a SNP, SNP it's a genetic variant, the environment is complex because we are testing all of them against the phenotype. And most of the time, like for instance, we are testing, like we are selecting the highest p-value one, so the most probable one, but there are other variants that are actually associated. So who is the causal variant here? And then many times, these variants are laying on a non-coding region of the genome, so we don't know actually what the variants are doing. And what are the effects that they have on neighboring genes. So for instance, in this locus here, we have this variant, but there are like 3 functional element genes, but we have also regulatory element in ACER, CTCF, because the regulation of the gene expression is very difficult. Oops. No. And we will never know the last, probably.
You can use the arrows of the keyboard.
Okay.
Or just click on the slide, because sometimes the slide gets unselected.
That was well. Okay, it appeared. Okay. And yeah, the effect on the downstream pathways. That also is where networks are coming into play. So this is a real scenario. So we zoom in a locus. And then we see that, OK, there is this NIB, that is the one that from genome-wide association studies we detected, but there are like other 10 significant ones. So, and in the area, there's a lot of genes, so what is the functional element here? Many times, since now, genome-wide association studies took the closest gene if the variant was not in a coding region, but we can do better. Because here it's changing, huh? But there no. OK. So this is where our first publication that is published yesterday, was published yesterday. We got the acceptance. We developed a library, a Python library for the unraveling of complex variant related to, for variants related to complex traits. So this is a very quick summary of what the library can do. So we can retrieve variants associated to a complex trait, defined as an experimental factor ontology. And then through the library, we can retrieve data from different sources in order to make sense out of the variant's consequences. So we can inspect the genome, we can get LD information. LD information, I forgot to mention, linkage disequilibrium is the phenomena for which we have many variants that are associated to the lower p-value variant. So this is the problem, because our variants are very correlated between each other. So we can see what are the regulatory elements in the surrounding areas. We can get QTL data. So QTLs are genes that are differentially expressed given that specific variant. So they tell us, OK, this variant is an encoding region, but was associated to a differential expression of this particular gene. It tells us about the effect of the variant. We can get the LD matrix, etc. So most importantly, we can also use the open target locus to gene pipeline. It's a very complex pipeline in which many analyses and data were implemented in order to make sense out of the functional effect of a specific variant. And they build an open target, they build this pipeline, but we can use it through Genopic in a Python environment. So these are how the variant is working. So here we run variant effect predictor, so a tool from Ensemble. So we have this snippet of code. So this is the function, and variance is just a list of SNPs. And then it's processed and gives us like this, I don't know, beautiful pie chart with our consequences here. So in order to have a glance on which variant we have. Also, we can get the LD metrics or the variants that are in linkage disequilibrium through a specific one to see the associated variants. EQTL, so having that specific variant affects the expression of a given gene. So it gives us glances on the function. And as I was saying, the locus to gene pipeline, so this was a publication from the Open Target, and it is very interesting. So what they've done, basically, it's taking all the variants from GWAS catalog, and then they perform a lot of analysis on this set. The lot of analysis is fine mapping, understand in the locus which is the causal variant. And then they integrated different type of data here. So they integrated epigenomic data, distance from the variant to the gene. They perform colocalization analysis with EQTL data. So we have understand if the variant is truly affecting the expression of the gene or is an artifact. They collect all of this data. They build a machine learning model that use this data here as features. And then we can run this model. They built a training set. So then they train the model, and we can use the model to give a variant, understand which is the functional element, and it assigning a score to the genes. So since my research revolves around intervertebral disc degeneration, we use Genopic. We use a locus to gene pipeline through Genopic to understand the effects of variants associated to intervertebral disc degeneration from a genome-wide association study. So here, these are the same plot, basically. They tells us the same thing. It's a function enrichment analysis. So on the left, it's a function enrichment analysis on the variants that without mapping, without Genopic. And we can see that there's a lot of autoimmunity functions enriched in the set of variants. This is the p-value, so the significance. In here, we have the same plot, but cooler, because made a network. So in the nodes, we have the functions. In the edges, we can see how many genes are shared between the functions. And the thickness of the edges are reflecting how many genes are shared. And the size of the nodes, it's how many genes I have for every function. And we can see that the function enrichment, and I don't know if you can read, it's very small, but these are transcription factors that are widely reported in literature to be associated to this degeneration. So we are shifting the attention of the set of genes related to variants from GWAS, from an autoimmune to a more extracellular matrix turnover set of functions. And this was our first publication. But genome-wide association studies are very expensive to do because we need genetic data that usually is protected and very big, and we need to pass through a data transfer agreement and many other things. And so there's a lot of tools now in the last years that try to associate genes to phenotypes in a more data-driven approach. And this is what we've done for our second project. So we build a role, like we build a niche, like we inserted us in this niche of research. And with data-driven approach, the problem Natasha presented that we have a lot of different types of data. So we have transcriptomics, proteomics, epigenomics. Every data is produced with different approaches, with different conditions, different normalization. It's very hard. So we need, and also there's not a gold standard model of integrating this type of data. So the model that we decided to use, how to model this complexity, was with networks. So we have many different networks in biology. So we have a homogeneous network, like a protein-protein interaction network, so in which nodes are of the same type, and connections also. So we can have two proteins and an edge if the proteins are interacting. And then we have multi-layer network. These are kind of the same. Here we have the same node, but different edges, different type of edges. So like two proteins here, like one interaction, another type of edges activation, for instance. Then we have multi-layer network, in which we have different type of connections. So from, like, we could have genes and proteins and functions and different type of edges. And then the complexity, the knowledge graphs. Sorry if I cite you, because we call them knowledge graph. And in which we have direction in the connection. We have many type of different nodes and many type of interactions. And this is probably the case that better reflect biology. So the technology that we used were knowledge graph embeddings. So having this knowledge graph here that is very, as also Natasha anticipated, is very close to natural language processing. We have here, there's a lot of biological information, and then we extract, applying algorithms, we extract embeddings. So we try to translate the knowledge that's inside here to a vectorial space that is of defined length. So they can have like 50, can be 50 length, like 100 length. We can decide that. And this is our second project. So which data went inside the knowledge graph? Ontologies, we integrated gene ontology, human phenotype ontology, disease ontology, protein-protein interaction data, drug target data. Also, we annotated like proteins to the functions of gene ontology, so which function is related to that, which protein is related to a specific function. And we end up with a very big knowledge graph. And this is an example. So from my IDD, intervertebral disc degeneration, like TGF-beta is a protein that is annotated to have a role in intervertebral disc degeneration. But then we have molecular data, so different scales. So interact with like directions and type of edges, and also bone morphogenic protein 1, like the function that is annotated too. So in blue we have proteins, here we have diseases, and here we have functions in gray. It's very simple. You have 1 million edges, that graph. And then, like the method, because we have we use the graph embedding, so the last topic covered by Natasha, and also here there are many type of methods that are applied to knowledge graph to transform it into embedding, to transform the nodes into embedding. There's graph neural network. Probably the most efficient one is relational graph neural network. Because we have a complexity, a high complexity. Then we have like normal neural networks. This is Trans-C. There's a ton of different algorithms and reworking on the same algorithms. So graph neural network, you feed the graph, and you aggregate toward neighborhoods of the nodes, and then the information is passed through the edges. So so we can encode the information inside the of the neighborhood inside every node. Then this information is passed through a neural network, so non-linearity, and then we obtain node embeddings. Neural networks, these are different type of models. So for instance, this one tries to minimize, it says that the embedding of the head, so node 1, plus the embedding of the relation, must be similar to the embedding of the tail. So for instance, to take a real-world example, Gioconda is in, and then, so I have Gioconda here, head is in, is the relation, tail is Paris. And so they're optimizing this function to obtain the embedding. But every model has its own function. Then we have the random walk plus and language model, and I should already explain, and a matrix factorization we can use then also for obtaining an embedding. I know how to do it now. Yes. So also very quickly, we developed two novel algorithms. This is a random walker plus a natural language processing method. So we theorized that in biology we have a defined chain of events. So for instance, to develop a disease, you can think that a protein is affecting, so a dysregulated protein is dysregulating a pathway that then is leading to the disease. So we have this chain of events here. Or the action of a drug. So a drug is affecting a protein that is affecting a function that is developing the disease. So we developed this algorithm in which you can assign weight to the next jump. So for instance, if we are in a starting point, we have different type of nodes, like medication, proteins, etc. And we want to jump to a protein with with a higher probability. We can assign more weight on the edges that go to the protein. And we made, and this is the formula that binds the weight that we assign to the probability. And we made like some simulation, and apparently it's working. And it was working well also, we'll show you the result. And of course, it's really dependent on the number of edges that leads us to a specific class, because we can have one edge that goes to a protein and 5 or 10 that goes to functions. And then we developed a shallow neural network also to transform the knowledge graph in embeddings. So briefly, we have here the relationship is not taken into account. So for instance, we have two proteins that are interacting. The proteins are mapped to an embedding layer that then is this embedding layer. So the two embeddings are selected. These are random numbers. We make the dot product of this random number. That is our link. We represent it as a link. And then we get a number, a score. And then we have labels to train the model. If the number is higher than 0.5, it means that the link is true. In this case, the link is predicted to be false because it's 0.2, but the label is a 1, so the link it's true. So the model is wrong and is training, is calculating the error and go back and update the weights of the embedding layer. So in this way, we have the embeddings. This is my actually second publication, but here it's also second here, so everything's fine. In which we made a work in which we tested this technology. So we built a knowledge graph. We created embeddings, and then we had two different tasks. One that was unsupervised, so we calculate the quality of the cluster, how well they are clusterizing, and one supervised one, so in which we try to, we took this genet and we tried to predict, try to test how well these embeddings were predicting disease association. We have GDI representation here because we need a representation of a gene and a disease to make the association. So we try different representation. It can be concatenated to embeddings of gene and the disease, summing them, or making the product, or the mean. And then we use them, we tested them with different algorithms, logistic regression, support machine, and et cetera, like the canonical algorithms. Made a grid search, and then we evaluate that on 20% of this genet that we took also out from the knowledge graph. So this was never seen by the model, because many times you can have leaking of information between the knowledge graph and the embedding creation step. This is how bad the embedding were clustering. But we can we can see that there is some clustering, because like, for instance, RGCN, there's no clustering at all. But like from a side point of view, we can see that metapath-VEC, maybe it's one that separates more the classes. So these are classes from Human Protein Atlas, so biology classes, biological classes. And this is for protein, and this is from disease, using ICD-9 medical vocabulary. So diseases are very difficult to clusterize because, like, define a disease itself, it's very hard. We see in the example for intervertebral disc degeneration. So we calculate the quality of the cluster, calculating the homogeneity score. It is a measure of entropy, basically. So how many elements of the same type are inside every cluster. And we saw that metapath-VEC was the higher scoring one, both for genes and disease. This is tailored for knowledge graphs. But ours was the second, so DLEM was the second. Actually, funny story. First, we submitted the paper without trying metapath-VEC, and ours was the first one, of course. But then, second round of revision, they said, "Well, why don't you try metapath-VEC?" And yeah, they performed better. And metapath-VEC was performing very well also in the supervised task. So it was the best algorithm. And we can see our BioKG2VEC is the second one also here. But we won for speed. We were faster. So our algorithm are faster than metapath-VEC, at least. So they perform more or less the same, but they are faster. So we measure also the speed of execution. This is a normal computer, i7. Detected the best model. We made a tool out of it using the best performing one. And we ran it on intervertebral disc degeneration. That is the aim. So these are the top 10 scoring genes, like the tool gave us as an output. And we see that the best scoring one is TGF-beta1, and we have SMAD3 that is acting in connection with TGF-beta1. And there are like other famous players in the disease, such as metalloproteinases and collagen, but also like LOX that are genes that are less studied. There was Spark also, but they are already reported in literature that can have a role in animal models and in vitro models. And the curious thing is that so this was the training set for this degeneration, so we had very few association in this genet, but 445 genes were predicted to be associated from the model. So only out of 3, so the model is learning from other, probably function involvement, etc. And these are scored as positive as associated, but are very lower in the ranking. So the model score higher genes that were not in the training set, basically. It's a cool sign. And then this is a function enrichment analysis. You already saw this before. You can make it with Genopic, this graph, if you want. It's to visualize. And yeah, so the function are extracellular matrix turnover and very related to the disease, relevant. And so it's a positive sign. But yeah, for sure, as we would need to validate them in lab. So we can conclude that we develop a knowledge graph embedding framework to prioritize gene involving complex conditions. The top scoring gene for intervertebral disc degeneration are reported in literature and already from the function enrichment analysis related to function relevant to the disease. And also integrating heterogeneous data, as we saw with Genopic with the first presentation, the first publication, we can shift like the attention to probably more related pathways through pathways in complex conditions. So we can focus the attention on maybe the right cause. And this was my last slide. Thank you very much. I'm happy to take questions. This was the first summer school I participated to. We're there, here in UPF campus. Thank you very much.
Any questions? No.
Can start, unless you want to start as a chairwoman. Yeah, thank you, Francesco, for that. So the question I have, so basically then at the end of the day, so you have your list of prioritized genes that are associated with multiple functions, pro-anabolic, sometimes pro-catabolic, depending on what is happening. And when you're doing your verification against the phenotypes, so are you making the difference among different phenotypes? Because I've shown gene degeneration and herniation. These are really two different phenotypes. So one is a degenerated one, and the other one is a herniated one. Mm-hmm. So you might have degeneration without herniation, and actually you might also have herniation with very slight degeneration. So basically there are three phenotypes that are important: modic changes, mm-hmm. degeneration, and herniation. So have you tried to make that exercise in differentiating the three?
So in the predictions, you mean like when we run the model?
Yeah.
No, we didn't try, but, and this is a very hot topic for these approaches, because actually we have phenotypes as experimental factor ontologies. So with an ID, no? And the fact is that when you then go to patient-specific data, you have a huge amount of different phenotype that changes from a small bit, like and with experimental factor ontology, we have this degeneration, and that's it.
Yeah, that's a bit my point, is whether the ontology is really correct. Because, yeah. For example, in one of the secondary genes that you have in the prioritization, you have HTRA1.
A1, yeah.
And this one has been very recently shown. It has been presented last week
at
ICELS. It has been shown to be associated with modic changes because of the innate immune system in interaction with this system.
Yeah.
So, but of course, if you put this degeneration as a keyword, you would have a low score, because the real keyword is modic changes, and modic changes is recognized as an alteration of the bone, not as an alteration of the disc. So now there is a shift of paradigm, where now we tend to associate that with the disc, but there's still a bias.
Yeah, for sure. But the bias is inside the data itself that we use. For instance, that one that I showed, the HTRA1, was associated to this degeneration in this genet, so it was in the training set. So it went inside the knowledge graph, but since the beginning, because when the author associated to this degeneration, not to modic change, maybe this was associated to modic change itself, but the author intended a phenotype as this degeneration, like taking into account also modic changes, and merge the phenotype modic change inside this degeneration. I guess we would need to go to endophenotypes, to split the disease in many different phenotypes and associate every different phenotype to its own biological pathway. I was reading some publication recently, and there's an approach that they are doing, because probably the phenotype, the molecular pathway that leads to modic changes are different compared to the one that leads to this degeneration. But many times, for absence of data, they consider this degeneration as a set of phenotypes, and that they call this degeneration to have a bigger sample size. Basically, I've seen this in London many times. So you have a bigger sample size. You can run more powerful studies. Probably in the future, we are going toward that direction now. We need to fine-tune every different phenotype.
Yeah, and so rework then the phenotype ontologies similarly to what Francis now is doing, I guess.
Yeah, exactly.
And integrating that in this. So how challenging is that? It is to integrate then these new ontologies, for example, in this genet?
Well, we make it from from scratch. We can remake it, but yeah, I guess it will take years probably of work, and it would take years of work in a like thought manner. No, like in this way, like all the people that is working in the clinical environment should work in the same direction. This is challenging. Well, it was challenging itself from the beginning of science, probably. Okay, so thank you.
Other questions? Sofia?
Hi, thank you, Francesco. Well, I have a question. Like you say that you have like your nodes or the proteins, and then you assign them embeddings, and you see like the performance of your model. I mean, is there I don't know, maybe that you get like a good score, but in the biological point of view, how can you know if this is reliable, even if you get a good score or not? I don't know. How can you test this, or how can you assess this?
Yeah, one drawback of these methods, and really depending on which method are using, it's that they're not as interpretable as for instance, matrix factorization methods, because of the deep neural networks, or the non-linearity. And so it's the neural network that is training himself and saying, oh no, this is wrong. We have the error, and so we minimize this error, but we don't know how. Probably the reason is the network topology. But like for instance, the random walk approaches are very interpretable, so you could use it. So you have the embeddings, and you can trace back the random walk. And you have like all the nodes that were visited, and then you can for instance say like this word is appearing many times, like this pathway here. So the random walk visited like this pathway many times. So there's a high probability that the embedding was created due to that, to this biological niche. Mm-hmm. You can trace back the walks, yeah.
Oh, okay. You will tell me how to do it. Okay. Thank you.
More questions?
Thanks, Francesco. Could you go back to the slides where you show the results and the performance?
Which one? The performance?
The performance of different models. Yeah.
This one?
The one. The next one. Yeah.
Yeah.
So how do you I assume that you have training set and test set to score the performance of these models.
So how did I measure it?
Yeah, do you have like to train the models, do you use the same batch size or learning rates, and are they same for all the models, or do you have to change them?
So I use the same number of epoch in all of them, and then I made 10 experiments in which, so the x-axis are the experiment, in which I sample like 100 random nodes from the network, and then with the edges. And then I created the embeddings, and I was calculating the time of the process with the computer. So we made it in the same way. So also, the philosophy behind the three algorithms is different. So we just set the number of epochs, but there are like a ton of parameters that you could tune and check. Also, the length of the embedding were the same. Mm-hmm. So we would try to be as much reliable, but having the same parameters as possible. So we sampled 100 nodes, ran the embeddings, and measured the time for all of the three in the same computer. This is.
So this is during the training time that you plot?
Yes, it's a training. Like the embedding is a training time. Yeah.
And do you run them on the GPUs or?
This is. Not GPU, CPU.
CPU. Okay.
Yeah.
Okay, thanks.
Yeah, there's not a lot of data.
Anyone else? Oh, looks like we are done with the questions. Thank you, Francesco.
Thank you.